Hello, in this video we will discuss about the hypersensitivity types. So in this video we will just discuss about the two types, and next video we will discuss two types of 3 and 4 hypersensitivity types. So basically the hypersensitivity, what is this? Basically, it is undesirable reaction produced by normal immune system, including allergies, autoimmunity. So the autoimmune disorder can be occur, and they are usually referred to as an overreactions, may be damaged and uncomfortable reaction. Also, it means the allos means the other and erga means the reaction. It means it will be damage the tissue, so that is the inflammation will be produced. So let's begin to understand the four types and what is the basic differentiation between the hypersensitivity types. So the type 1, type 2 and type 3. Here I am drawing. And in this way, the type 1 hypersensitivity involves the IgG, immunoglobulin G antibodies basically. It is the mediated and immediate a reaction. Example is the pollen. So the pollen allergy is due to the type 1 hypersensitivity. While the type 2 is the antibody mediated reaction, IgG and IgM involving this process, and the antibody is the cytotoxics and non-cytotoxics. So the 3 type is the immune complex mediated reaction, while the 4 is the cytotoxic cell mediated delayed hypersensitivity reaction. So in this way, two types we will discuss in this video, while the two type we will discuss in the next video I will make, and I will give the link in the description below. So the type 1 hypersensitivity is involves the immediate reaction. In this way, the pollen involved. So the pollination will be increased, the seasonal pollination due to the increase of the carbon dioxide. Industrialization will lead to increase more carbon dioxide with the passage of years. And that's why the photosynthesis will increase due to the increase of the carbon dioxide, because it is a greenhouse gases. And the pollination will increase, mean the pollen grains will be increased due to the cross fertilization and the cross pollination. So in this way, the pollen allergens which that will move during the breathe inside is used. So it will infected person inhale this pollen, and in this way the macrophage are present in the lungs epithelial cell, and macrophage are present in the alveolar sac. So in this way, the macrophage will recognize that via a toll-like receptor this allergen and will endocytose. After the endocytosis, the macrophage will go as an antigen presenting cell and recognize via a MHC class 2 molecule via a T helper type 2 cell, which contains CD4 molecule, is a naive, and which that is basically is the primary lymphoid tissue, which that will move toward the primary lymphoid tissue. And in this way, macrophage is also produce the interleukin 4, 5 and 10 and cytokines. So in this way, the recognition through CD4 to produce to maturation of this T cell, and after this maturation, it is also recruited via interleukin 10 from the releasing from the macrophage, which that will attract the eosinophil, and the both function are same, macrophage and eosinophil. While on the other hand, here is the T cell activation will lead to recognize the B cell antigen, piece of antigen, to lead to increase the plasma cell, and the plasma memory cell and B memory cell will lead to produce the antibodies, and these antibodies will be bound with the Fc receptor with the mast cell. After the binding with the Fc receptor, also the allergen will also bind with the Fc receptor. So in this way, the mast cell basically release the pro-inflammatory mediators like histamine, tryptase, serotonin, and heparin. So these three types of the allergen, three type of the inflammatory mediators will produce, and it will target the tissue. For example, here it will bind with the receptor, histamine receptor, to trigger the pathway, the phosphorylation pathway, and in this way the necrotic factor kappa B will be produced and work autocrinally or paracrinally. So in this way, after the binding with the necrotic factor kappa B, we are receptor, necrotic factor kappa B receptor or death receptor. In this way, fast ligand, as a fast ligand, the fast receptor bind, and the apoptosis will be occur via this tissue damage. So it can be, in fact, the damaged tissue, also the non-infected tissue also. So basically, it is the tissue is not damaged, but damaged through a necrotic factor kappa B. So the apoptosis will be occur and cell death lead to increase the inflammation more. So the type of allergen can be for food, nuts, eggs, soy, wheat, animal, bee, wasp, cats, insects, and rats. While the abiotic can be dust, latex, and pollen. Basically, pollen is also abiotic because it is inactive without fertilization. So the type 2 hypersensitivity, which that is a separate, and in this way involve the immunoglobulin G and immunoglobulin M, are directed against cellular or extracellular matrix, antigen resistant, and cellular destruction and damaged to tissue. So the tissue will be damaged. And in this way, the cytotoxic mechanism 1 and cytotoxic mechanism 2, 3, and 4, which that is involve the different pathway we will discuss in this manner. So the hypersensitivity type 2, which that involve the cytotoxic mechanism 1. In this way, here is the non-self and self antigen. For example, the self antigen is our tissue, while the non-self antigen is can be bacteria or other things or virus. So in this way, here is the infected self antigen. If so, it will be CD4, which that are present in the primary lymphoid tissue and reside in the primary lymphoid tissue, and B cell, B naive B cell and naive CD4 T cell. And in this way, CD4 T cell, which that will be recognized and through a non-self antigen recognition as well as self antigen recognition, both can be occur. So here is a bone marrow, which that's contain a B cell maturation. After the maturation, it still naive and go into the primary lymphoid tissue, and this B cell basically after the recognition through CD4 T lymphocyte recognized via a B cell receptor, a B cell here you can see, and after this the piece of antigen will be recognized through antigen representing cell through CD4. After the recognition, the B cell activation and to lead to produce the plasma cell after the proliferation, and the proliferation will lead to self antigen. If the infected our own cell will lead to self antigen, and the infected cell will be killed obviously, but the antibodies production self reactive and other similar tissue damage by opsonization, and these can be healthy cells. So it means the autoimmune disorder can be occur, while here the non self reactive is positive and it is helpful, while the infected self antigen is harmful because it will infect to others. For example, here is I am giving the example. For example, here is the RBC. RBC is the antigen antibody complex with a host tissue problem. In this way, the immunoglobulin G will bind with the antigen of the RBC, as well as here is the antigen, which that is the foreign particle or foreign bacteria, which the antigen antibody complex non-host tissue infection. So in this way, that will be opsonize and kill that pathogen, while our own RBC will also lysis due to the autoimmunity reaction. So this is the cytotoxic mechanism 1. In this way, the example of this basically is the hemolytic anemia will be caused due to the cytotoxic mechanism 1, because the cyto means the cell and toxic means the poisonous. So here is the complement system also involved for the cytotoxic mechanism 1, which that is can be small proteins which that come from the liver and synthesis in the liver, and enzyme cascade, as well as the fight infection. And in this way here you can see this is a cleavage formation. After this recognition with the via antigen antibody complexes which that bind with the Fc portion of the antibody with C1 protein will lead to trigger the C2 and C4 C3 to C9, and this cleavage formation will lead to activate the protein after the cleavage to create a C3A, 4A and 5A. That is the chemoattractive factor. So this chemoattractive factor will recruit the neutrophil, and this neutrophil will lead to trigger the production of enzymes. Enzyme can be after the degranulation. Enzyme can be peroxidase, proteinase 3, myeloperoxidase, and degranulation will be occurring in this manner. And reactive oxygen species will be produced due to this enzyme, and this is the neutrophil will be decreased due to the rupture of the neutrophil. So remember about that, the neutrophil will be decreased, so the neutropenia will be occur, and in this way the thrombocytopenia will be occur because the thrombus formation will be occur, the RBC clot formation will be occur, and the hemolytic anemia is also occur. Hemolytic means that RBC will be ruptured due to the reaction of this smaller protein, that is the C protein, which that is complementary protein, and that is known as the complement system, which that is involved from synthesis from the liver. So the tissue will be damaged through a reactive oxygen species also produced. So in this way, I hope you make sense. Let's begin to understand the cytotoxic mechanism. Second, the two cytotoxic mechanism. In this way, remember, this is the circulatory system which that contains C5b. C5b is the basically is the protein after the cleavage formation of this cascade of the cleavage formation will lead to trigger the C6, 7, 8 and 9. After the 9 triggering, the 9 one by one will bind together to form a membrane attack complex formation. So the membrane attack complex formation will lead to this bind with the membrane of the RBC. After the binding with the membrane with MAC, that is the membrane attack complex formation. So in this way the MAC will bind with the RBC, will function is look like this. I am drawing here, you can understand. And in this way here is the outer cellular matrix and inner cellular matrix. Outer contain different osmolarity while the inside is different, so it is important for the survival and homeostasis of the cell. So the disrupt of the homeostasis due to the binding of the membrane attack complex. After the membrane attack complex bind to move the albumin inside into the cell more as compared to the extracellular albumin. Because the albumin contain negative charge, so it will also attract other sodium and potassium as well as the water, because the water will attract through a sodium and potassium. So in this way the water will fill more, and the osmotic gradients will be occur inside, more concentration of the solute and solvent while the outside. In this way you can understand that will be cell lysis and burst the cell. So that is known as the hemolytic anemia also. While the cytotoxic mechanism, the complement system involved in the next here is the cytotoxic mechanism 3. In this way, the cytotoxic mechanism 3 involves the complement system more importantly. And in this way, here is the RBC, which that is bind with the immunoglobulin, and the FC portion will bind with the C3b and C3b complement protein here. So the C3b complement protein after the cleavage formation due to the cascade of the cleavage will lead to recognize in the spleen. So it will go into the spleen, and the macrophage will filter this opsonized antibody antigen complex, and it will recognize via a C3b receptor or only Fc receptor via a macrophage and neutrophil. So it will destroy by macrophage and neutrophil. And neutrophil also produce the inflammatory mediators which that will attract more neutrophil and more macrophage. So the inflammation will be caused. So the cytotoxic mechanism 4 is involved, the antibody dependent cell mediated cytotoxicity, ADCC. And in this way the natural killer cell which that is involved in this mechanism, and the natural killer cell will recognize the Fc portion of the antibody with the antibody in antigen complex formation will lead to a cell lysis. In this way the cell lysis mean because the complex formation the natural killer cell will kill the plasma membrane. So the plasma membrane of the RBC will be killed and damaged, lead to exocytosis or efflux of the nutrients and etc. and protein will lead to cell lysis. So this is known as the bursting of the cell. While the non-cytotoxic mechanism which that involve the part of hypersensitivity type 2, which that is the antibody mediated cellular dysfunction. And in this way non-cytotoxic, it means the cyto means cell and toxic means the poison, so cell will not damage, while the inhibit via a receptor, and receptor will be inhibit through a antibody inhibition through a recognition from a CD4 lymphocyte. The helper cell will recognize, and in this way the activation and ligand will not bind because the collapse this receptor as an antigen recognition. For example, the major example of the Graves' disease. In this way, the nerve impulse will release a neurotransmitter, will bind with the neurotransmitter receptor, and it means it will not bind because the neurotransmitter receptor will be against the as an antigen will bind with the antibodies. So it will be autoimmune disorder. Auto mean itself. Immune mean the immunity. Disorder mean the dysfunction. So that means the non-functional neuromuscular junction will lead to movement, breathing, and there are a lot of complications during the speaking as well as during the urination and everything. So I hope you make sense about the hypersensitivity type 1 and 2. Please make sure to subscribe.